Welcome, everyone. Thank you for joining us for today's webinar, Nanostructured Molecularly Imprinted Polymers, Robust, Stable, and Selective Alternatives to Antibodies. It is presented by Adrian Kinkaid, PhD, the CEO of MIP Diagnostics. My name is Judy O'Rourke, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Kinkaid. I will now turn the presentation over to him.
Thank you very much for that introduction and also the invitation to give this presentation. So not many people have heard of molecularly imprinted polymers, which is a bit of a mouthful, so we tend to call them MIPs, and certainly not the form of MIPs which I'll be speaking about today, which are nanostructured molecularly imprinted polymers, or NanoMIPs. So just to cover some of the different versions of MIPs which exist, the original format was what we now call bulk MIPs. These were originally invented around, I think it was 1930, so they've been around for a very long time. And they're formed by a self-assembly of monomers, which form around a mixture of the target or template molecule, and are then polymerized in situ to form a bulk polymer with the target molecules embedded within in a kind of honeycomb style. However, they're very difficult to use for all but a few applications, and what would be much more applicable would be something where you could use it much more like antibody, in other words, a polymer antibody. And this is where NanoMIPs come into play. So NanoMIPs are discrete nanoparticles containing just one binding site per particle. They can be manufactured in different sizes, but the typical range for us is between 30 and 150 nanometers in diameter. So slightly larger than your traditional protein antibody. We can actually make them much smaller than that. And we can make them so small that they become linear in structure and are more like a chemical aptamer. So these can be sort of 1 to 3 kilodaltons in length. And they're starting then to become more drug-like than actually polymer-like. The other form that's on this slide is what we call RDPs, or rationally designed polymers. These can actually be made using the monomers without any template molecule. So technically they're not imprinted molecules, and I'll not go into any more detail on them today. But our proprietary technology focuses on a novel method to make NanoMIPs, which circumvents a lot of the drawbacks associated with the traditional MIP manufacturing methods. And the resultant NanoMIPs are very much like plastic antibodies, but are very easy to produce and are incredibly robust in comparison. So some of the main benefits of using MIPs as compared to antibodies. Well, if we start with antibodies, they have evolved to be stable in physiological conditions, pH 7.4 or thereabouts, body temperatures, whatever they may be. But they are relatively expensive to produce, and being proteins and therefore built up from the 20 amino acids, There is some limitations to the chemistry that's available to build an antibody. And of course, the scaffold also determines the surface area of the interacting variable regions. Being proteins, they can be difficult to integrate into sensors and certain assays, and certainly couldn't be used, for example, in organic phase. MIPs, on the other hand, are very stable in harsh conditions. We can use them in organic solvents. We can wash them with 1 molar hydrochloric acid, pH 1, and they'll still survive. They can be made to work in seawater, for example, or even in urine samples. They're incredibly robust. They can be made to bind at 80 degrees centigrade, and indeed they can be autoclaved at 121 degrees centigrade and still retain nanomolar affinity for their target after the process. They can be prepared for any target. There's no immune response required. We don't use animals at all, but they can be used for any target, from very small molecules, drugs, or smaller metabolites even, right the way up to through whole peptides, whole proteins, and even viruses. They are incredibly easy to functionalize because we can build in functional groups for you. We have complete control over the chemistry of their composition. There are around 5,000 monomers available for us to select from. They've all been used in the literature. So much more chemically diverse than the 20 amino acids available to proteins. They're very fast in terms of development. Typical development project for us is about 6 weeks. And in terms of manufacturing, well, that process in itself takes about 4 hours. So what kinds of MIPs have we produced? Against pretty much every target, whether they're pharmaceutical drugs or drugs of abuse, metabolites, toxins, which may be a threat to food security, proteins for studying drug targets or other assays, biomarkers, and diagnostics. We can produce them, as I mentioned earlier, against whole cells or viruses, bacteriophages as a surrogate for viruses as well. Man-made toxins such as pesticides, and indeed explosives for homeland security applications. So we are not limited to those targets which would elicit an immune response. Small molecule drugs are used commonly because they don't elicit an immune response. And also hosts, proteins, should also not elicit an immune response in that host. Therefore, very similar proteins are also difficult to get antibodies to. None of these are issues for us. Some of the MIPs that we've developed in their applications, we've got customers and run projects in a wide range of application areas, including drug discovery, but also diagnostics, separations such as bioprocessing, and also bioanalysis. We've done this for a number of different companies across a range of industries. If I take you through the manufacturing process to make NanoMIPs, you're getting a better understanding as to how they can be made and what they can be made against. So let me take you through our proprietary methodology. Well, first of all, we need to understand what the project needs are, or what the customer needs are. We then design the monomer library that we're going to use for that particular target, selecting monomers with a propensity to bind to the target. This gives us the option to target a given binding site as well. So if you had, for example, a cofactor binding to a protein, an enzyme, we can mimic that with a monomer and bind to the same site. If you had a drug that bound to a known site, we could target that by using similar chemistries to make monomers that are similar to those and build MIPs around that site. Computational chemistry can be used, and indeed we hold a patent on the selection of monomers for in silico for further development into MIPs. Once we get into the manufacturing method, we use our proprietary solid phase synthesis, which I'll explain in a bit more detail later. But importantly, we also use controlled polymerization. And again, I'll take you more through this I'll take you through this in more detail. We can add functional groups either to the monomer mix in terms of the bulk of the polymer as it's made, or indeed we could coat or add functionality to the outer surface of a MIP once made. We then need to make sure that it works in the way that we want to. Typically that involves affinity for the given target, possibly affinity against or lack of affinity for a selectivity target. Only then will we ship NanoMIPs to the customer or partner for use in their labs. It may seem like quite a lot, but the whole development process takes about 6 weeks or so. And in comparison to making antibodies, well, they can take up to 9 or even 10 months to manufacture, depending on the type of antibody being used. So let's take a closer look at the monomers. These are our building blocks and the equivalent of amino acids which make up proteins such as antibodies. So as I mentioned earlier, there's 5,000 different monomers available in the literature already that have already been used for making MIPs of various forms. There are equivalents to each of the functional groups or side chains of amino acids within the repertoire of monomers that we can use. And there's a host of other chemistries as well that you will not find in proteins. This gives us a lot more diversity and enables us to make sure that we get the best tool for the job in making a nanomip. So the manufacturing process itself starts with a target molecule. Here we've got the ring structure of melamine, so the nitrogen-rich 6-membered ring that's attached to a long linker chain. And via that attachment, we immobilize it onto a solid support. So in this case, we're using glass beads. I should say nothing on this slide is for scale, but the idea is to get the message across as to how we make them. We can then introduce a selection of monomers, as shown at the top of the box here. And each monomer is able to bind to the target free in solution. This means that each interaction via a self-assembly process is actually optimized because each monomer will find its lowest energy state, its optimal binding position. And it's only when they're actually bound into those positions that we will then polymerize them in situ around the target molecule. This is done by using controlled polymerization, and we use UV light to activate a catalyst. That catalyst produces free radicals, and that creates the polymerization process. So that's represented here by the blue blobs, which are polymer and down to the active site. Again, nothing on this is to scale, it's just to get the idea across. There will be background polymerization and low affinity polymers formed, and we want to remove those from the mix. Well, because we've immobilized the target onto a solid support, we've effectively made an affinity column. So all we need to do is wash away the low affinity components. Once we do that, the higher affinity components are retained on the column, and we can increase the stringency of the wash or the elution process in order to harvest those high affinity MIPs. What you'll also notice is that in this process, we've regenerated the column, and it's possible to reuse a template molecule or reuse a column to make an additional batch of MIPs. It depends on how robust the template molecule itself is. If you had a fragile protein, it would be less easy to reuse it. But melamine, as in this example, we actually made 30 batches of NanoMIPs from the same column. The controlled polymerization is a key to making this. We want to get the polymerization to bind each of the monomers which are interacting with the template molecule into a single entity so that they work together and create a single binding site. But what we don't want to do is have each of these individual NanoMIPs or nanostructured molecularly imprinted polymers fusing together or getting too large. we want to be able to separate out each of those binding sites. So having discrete particles is essential. We do this using our particular living radical polymerization method, which relies, as I said earlier, on UV light illumination. When the UV light hits the catalyst, it creates the free radicals, which promotes the polymerization. However, when we turn the light off, the same catalyst now acts more like a vitamin D and mops up the free radicals, inhibiting the reaction and stopping the polymerization process. This slide just shows a cartoon to try and get that message across. It's not a genuine trace. This gives us the ability to mix in mix in fluorophores, for example, into the monomer mix. But it also gives us the opportunity to stop the polymerization, change the monomers, and add a coat, for example, of fluorophores. Making MIP fluorescent is relatively straightforward. Indeed, we can build MIPs around other particles preformed in the mix. So in the case of B panel here, it's an iron oxide nanoparticle, which is in itself encapsulated in the slightly lighter grey of the polymer MIP that we formed. This means that the MIPs themselves are now inherently magnetic. By including monomers with various functional groups, it enables us to attach other entities to the nanomip itself. So it's quite possible for us to add a fluorophore, a drug, a protein, a reporter group, peptide, biotin, free thiol groups for immobilization, or indeed PEG. So we have complete control over the chemistry and complete control over the nature of the nanomips produced. We can make them hydrophobic or hydrophilic, positively charged or negatively charged. Pretty much whatever size you need, and indeed we can add different colors to make them whatever color you want. How consistently can we do that? Well, we've been able to automate the process of making the nanomips. We have a machine which can take out the human error involved in moving from batch to batch. The data on this slide shows 6 independently synthesized batches of nanomips against the same target, in this case, MSQ bacteriophage, which is a surrogate for viruses. Not only can we see that the diameter of the nanomips produced is consistent between 205 and 238 nanometers in diameter, but also, and more importantly, the association constant for the target is between 1.1 and 3.3 nanomolar in 6 independent determinations. That's within experimental error. To all intents and purposes, the batches are behaving identically. So how do MIPs compare to the traditional antibodies which most life scientists have used? Well, it's rather controversial to generalize about antibodies. Similarly, it's quite controversial to generalize about MIPs. But if we gather the populations, then typically nanomips are equivalent in terms of their affinity to antibodies. They're high affinity. We also have the ability to make lower affinity nanomips if that's required for a project. You might want something to bind and then be released for separation, for example, or sensors in real time. Robustness, though, we find that nanomips are very, very different to proteins, including antibodies. So they can withstand exposure up to 121 degrees without denaturing. Because the backbone is a carbon, 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 carbon polyethylene chain, it's not biodegradable. So the shelf life could be tremendously long. It's also very, very chemically stable. Batch-to-batch variability, as I showed in the previous slide, is excellent and better than you would necessarily expect, even from a monoclonal, certainly better from different polyclonal productions from different animals. Shelf life, we don't know, but it's probably in excess of 6 years or so. Time to develop, instead of months, we're down to around 6 weeks. And the manufacturing time is about 4 hours as opposed to maybe 4 weeks for a batch of antibodies. Moving on, how do they compare in terms of assays? This slide shows 2 dot plots using different fluorescence tags on MIPs, and you can see they work in much the same way as antibodies. A comparison in Western blot, where the MIP is on the left-hand side and the antibody is on the right-hand side, targeting some degraded trypsin to show the different epitopes and the different bands that might be available. You can see virtually identical results between the antibody and the MIP. Potentially more importantly, the column for the lysate is showing virtually no signal whatsoever. It's very, very clean, demonstrating that the MIP has very little non-specific binding. Applications of various NanoMIPs include epitope mapping. This is a relatively novel development, but we can use a NanoMIP made against a particular binding site, so we can select monomers to target those particular sites to protect degradation of the target from trypsin digest, for example. Then by using mass spectrometry to determine the sequences of what's being protected by the MIP, we can determine where the MIP is binding. This is a very rapid process and allows us to get sequence data for individual epitopes in around 2 weeks. A project would take around maybe even a year to do with antibodies. We had to raise them for that process. So this is also extremely useful for hopping from a protein to a peptide, so that we can then make MIPs to a less expensive peptide for use on proteins at a later date. It also enables us, for example, to spot differences in epitopes on various viruses, so changing between viral strains, which can be very important for vaccines research. Detecting viruses and indeed removing them in water has been something which MIPs have been used for. So in this particular case, the published example where the batch-to-batch variation was described earlier, the intention was to remove viruses from drinking water to ensure it was safe. I've already talked about the dissociation constants being around nanomolar affinities. But what was also demonstrated was that we could remove 9 logs of clearance in terms of removing the viruses or MS2 bacteriophages as a surrogate. So that goes from 1 billion plaque-forming units to 1 in a single column step. MIPs are very useful for using in sensors. We can take NanoMIPs and put them into a film, attach that to a sensor, making them ideal for detection of materials in real-time flow, especially when combined with the ability to adjust the affinity for the targets there by determining or predetermining the affinity from the NanoMIP manufacturing process. We can use them to imaging on a cellular basis by picking up particular targets, whether they are membrane-bound proteins or other elements of a cell, or we can indeed use them in whole cell or whole animal imaging on a larger scale. Internalization is something which has been useful for drug delivery and drug discovery approaches. And in this particular case, we made MIPs to an oncology marker, EGFR2, and the MIP, once it bound to the receptor, was naturally internalized by endocytosis. By attaching drugs to the outer surface, in this case cytotoxic drugs, to the outer surface of the NanoMIPs, they can be released after internalization due to the change in the chemistry of being internalized. So a labile linker is used to deliver cargo to the cell. Interestingly, in this particular case, the NanoMIPs themselves, being non-biodegradable, find their way to the lysosome, and therefore it's possible to determine the dose on a cell delivered to a cell on a cell-by-cell basis, simply by counting up the spots, the punctate marks highlighted with the small arrows here, within the lysosome of a cell. And comparing the phenotype to the number of spots gives a dose-response correlation. So in summary, MIP Diagnostics offer access to NanoMIP technology, which can be used in a broad range of projects, including applications in drug discovery as alternative to antibodies, in diagnostics, separations, and bioanalysis. Proprietary NanoMIP design and development processes enable us to provide high-quality NanoMIPs, typically within 6 weeks of receiving the target, and they will be functionalized to suit the needs of that particular project. Excellent recognition properties have been achieved for small molecules, peptides, proteins, and indeed whole viruses. They can also be modified with various functionalities according to your needs to make them fluorescent or electrochemically active or magnetic. I should like to thank you very much for your attention during this talk, and any questions that you have, I'd be happy to answer via the email.
Thank you, Dr. Kinkaid, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through May 27th, 2019. As a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Goodbye.